We are quite new users of the Attune, and we have the Violet 6 configuration, and today I will talk about some tips to phenotype human B cells. So first of all, why we have to study B cells? Because as you all know, the cells are a critical arm of the immune system because they are able to produce antibodies and to provide us protection. But besides their capacity to produce antibodies, these cells has many other functions that are mainly antibody independent. So as already mentioned, they can provide memory through the differentiation of plasma cells and memory B cells, but they can do also antigen presentation to T cells and regulate the immune response through the production of many cytokines that can be also pro-inflammatory, such as IL-6 and TNF-alpha, but also immune suppressive cytokines such as IL-35, IL-10, TGF-beta, that are cytokines produced mainly by the regulatory B cell population that can also express surface markers that can dampen the immune responses such as PD-L1 and PD-1. And not least function is the capacity of B cells to induce the formation and to promote the formation of tertiary lymphoid structures that are important, for example, in the immune response against cancer. So in the last years, the role of B cells has become evident in many diseases that are not only infections or autoimmune disease. As you can see from this example, there are biomarkers related to B cells in Alzheimer's diseases or in cardiovascular diseases, and the role is currently being studied in also in relation to the progression of solid tumors. And for that reason, we can say that for many diseases, B cells can be potential biomarkers. But there are some problems with the counting and the characterization of B cells in literature, in my opinion, because in many paperwork there is a limited use of markers related to B cells, such as, for example, many studies claim that there is a prognostic significance for CD20 positive B cells, but CD20 is expressed on many B cell subsets. And there is also an imprecise extrapolation of the significance of some markers, and there is the forced inclusion of multiple B cell subsets in the heterogeneous population. So that it is summarized in this simple table. You can see that there are a lot of B cell subsets. For example, we have 3 types of transitional immature cell subset. We can differentiate naive B cells in resting, activating, energetic. We have a lot of type of memory B cells, but we have also some atypical, let's say, subset of B cells, such as the double negative B cells and the regulatory B cells. We have to mention the age-associated B cells. So there is an increased need to define and correctly characterize every B cell subset. Also because every subset represents a different stage of differentiation and has different function. So, as you all know, B cells derive from the bone marrow, where the mature B cells can exit to the bloodstream as a transitional immature B cells. It can develop into naive B cells. The naive B cells can migrate to secondary lymphoid organs, and if it finds the correct antigens, B cells can generate germinal center reaction and give rise to plasma cells or memory B cells. So we developed for the Attune NxT this, let's say, backbone panel to identify the main population representing the peripheral blood B cells. And you can broadly identify B cells by using, of course, the live and dead markers. You can use the CD19, that is a lineage marker. You can add the CD20, that is another lineage marker for B cells. But to differentiate the population, for sure you have to add the CD38, the CD24, and the CD27. And let's see why. So this is our basic gating strategy. So we select lymph nodes, single cells, living cells, and B cells, and then we plot the expression of CD24 and CD38. That, as you can see in the immunophenotype listed here, these two markers are expressed in unswitched memory, switched memories, transitional immature memory, and naive memory, but also on antibody secreting cells. So we need more markers to correctly put every subset in the correct population. And as you can see in this dot plot, there is no clear separation between all the population. Of course, there is also the plasma cells population, and we did not include in this backbone panel their characterization, because you need to add exclusion markers, such as for lymphocyte monocytes, and you have to add the CD138 marker. So the first step is to try to identify this four group, so the CD24 high, CD38 high population, the CD24 positive, CD38 low population, the naive population that is characterized by intermediate level of CD24 and CD38, and the antibody secreting population that is characterized only by the expression of CD38. To further characterize this population, you will need the CD27 that is plotted against the CD24. The CD27 is a characteristic of mainly memory B cells, but to confirm the identity of all these subsets, we use also the IgD and IgM expression, because the expression of these immunoglobulins is differential in all these subsets. So this is just an example to how to correctly identify and set the gate to study transitional immature and naive B cells. So for transitional immature, you have to put the gate on this quite little population that is that express high level of CD24 and CD38. You need to check that they are negative for CD27, and you need to check that they express a high level of IgM and IgD. The expression of IgM and IgD is the thing that distinguish them from the naive B cells that are just above them. They are still negative for the CD27 and also for the CD24. And as you can see from this dot plot, they express lower level of IgM and IgD. The third group that we can identify is the memory B cell subset that express higher level of CD24, but that has lower level of CD38, but more importantly is positive for this time for CD27. And as you can see from the expression of IgM and IgD, if we don't use these two markers, we are going to miss two different populations. In fact, we can distinguish the switched memory B cells that are negative both for IgD and IgM, and the unswitched memory that instead are positive for IgM and IgD. The last group that we can identify with this basic gating strategy is the antibody secreting cells that is negative for CD24 and positive for CD38. We can first check the expression of IgM and IgD. They are both negative for the expression of immunoglobulins. And we can distinguish two different populations according to the CD27 expression. Again, we can distinguish the various differentiation step of a plasmablast by checking CD20 expression this time. And as you can see, early plasmablasts have a higher expression of CD20, while plasmablasts almost completely lost the CD20 expression. So we applied our panel to the tumor infiltrating B cells, and the first question is why we need to study B cells in the context of solid tumors. Everybody study T cells, as you all know. Okay, so these are just potential role of B cells in tumors, because many of them have been not described well. So as you can see from the picture, B cells can of course produce a lot of cytokines and chemokines that can help to organize and recruit other immune cells to the tumor site. They can, of course, prime and shape the T cell response. They can activate through the production of antibodies the innate immunity, and they can have also a let's say not positive function of immune suppression made by regulatory B cells that can, in some cases and in some models, mouse models, have been demonstrated to improve and to promote cancer growth. So we applied the same panel to these are just an example of two different tumor infiltrating B cells in cancer. So we first we add, of course, to the panel the CD45 marker to identify the immune population, and we applied the same panel that I just already explained. But the problem is that in that case, the situation for the gating is even is difficult, okay? Because as you can, if you compare the gating strategy with for the peripheral blood, you cannot distinguish even the transitional that you don't want in your tumor infiltrating B cell population, because otherwise it means that maybe you have a contamination with the peripheral blood, but you cannot distinguish memory B cells and naive B cells. So you can, of course, put the same gate on your sample, but you need to check, of course, the expression of CD27, IgM, and IgD. And as you can see from the plot that I reported here, you can still distinguish the same population also between tumor infiltrating B cells. So you can nicely distinguish unswitched between memory switched B cells, and you can, of course, estimate the population of antibody secreting cells. So another very interesting population of B cells is the regulatory B cells that have been described in many pathological settings. For example, it has been described almost for the major part in a mouse model that can promote cancer development, but they can also prevent allograft rejection. They are involved in many autoimmune diseases, and they are present in a lower number in patients with autoimmune disease, such as multiple sclerosis and RA, okay? And their main function is to regulate, for example, the differentiation and the function of T cells. For example, they can suppress Th1, Th17 differentiation, but at the same time, they can promote the regulatory T cells population. They can also act on monocytes and also on NK. Well, but the main problem with these cells is that there are no unique surface marker to identify them. So you can choose your favorite, let's say, BREG subset, but there are no unique marker that allows you to identify this population, like, for example, FOXP3 for regulatory T cells. So the main markers that are used for these cells are their ability to produce immune suppressive cytokines such as IL-10, IL-35, and TGF-beta. And they have been described for this reason in many other subsets of B cells. As I highlighted here, you can find regulatory B cells among the immature or transitional B cells. You can find regulatory B cells among plasmablasts, as well as in memory B cells. And you can also have some different and more peculiar regulatory B cells, for example, these that express granzyme B. So how can we identify regulatory B cells? So there are many tips, and according to the literature, you can claim that your B cell subset is a regulatory B cell subset if they can in vitro suppress CD4 and CD8 T cell proliferation, if they can induce regulatory T cells, and if they can produce immune suppressive cytokines. So In the lab, we study mainly IL-10 producing B cells that can be quite easily obtained by a stimulation protocols that can be applied on PBMCs. You can just stimulate them for 5 hours with CpG, PMA, immunomycin, and the addition of preferred DNA to retain the IL-10 secretion inside the cells, do an intracellular staining, and then you can immunophenotype your cell. So the fundamental thing, as in all other experiments with the flow cytometer, is to put the correct controls. In fact, in our experiment, we put the FMO for IL-10 and also the unstimulated samples, because this allows us to set the correct gate for the IL-10 positivity. And as you can see there, in an healthy subject, you will find more or less the 2 to 5% of IL-10 positive B cells. If we have a look on their phenotype, you can see that there are no unique markers for their identification, although the IL-10 positive population express higher level of IgM, CD24, and CD27, but their expression is not unique to IL-10 positivity. In literature, you will find that also the CD9 is a regulatory B cell marker, but from my experiment, I do not see that thing. Maybe I'm using the wrong clone for the CD9, but what I can say you is that in the mice, instead, the CD9 works well to separate IL-10 positive from IL-10 negative B cells. So, if we have a look to the enrichment of B cells in the various B cell subset, okay, we can see that there is an enrichment in some B cell subset. This is the percentage of IL-10 positive B cells among all the CD19 population. And if you want to study the BRAG among the memory B cell subset, I would recommend you to use this gating strategy to select the CD24 positive, CD27 high positive population. And as you can see here, we have a slight enrichment of IL-10 positive B cells, okay, in this population that is much higher than the CD24, CD27 negative population. The other BRAG that are well studied in literature is the one that stays among the CD24 high, CD38 high transitional B cell population. And again, you can find an enrichment of these cells, but this population, the transitional one, does not fully represent the regulatory B cell subset. You can also, of course, expand your BRAGs in vitro, and the protocol that we use to obtain the 6 to 8% of IL-10 positive B cells after 72 hours of stimulation through the CD40, plus, of course, the addition of 5 hours of CpG, ionomycin, and PMA. And you can see that you reach a high number of regulatory B cells. And these cells are enriched again in the CD24, CD27 positive population. You will not find BRAG in the transitional immature B cell because after 72 hours of stimulation, you lose this population. So, which are my suggestions if you want to study BRAGs? So, if you want to just estimate the, let's say, ready regulatory B cells that you can find in your sample at steady state, you just have to take your sample and stimulate yourself for 5 hours of CpG, PMA, immunomycin, and you will find the percentage of regulatory B cells that are ready to function in your sample. Instead, if you want to see the potential of your, let's say, patients to produce regulatory B cells, you have to take your cells, stimulate them for at least 72 hours with anti-CD40 or CpG in some cases, then add the 5 hours CpG, PMA, immunomycin, and then you have the estimation of the potential regulatory B cell production of the cells that belongs to your sample. So I left you with our backbone panel, okay? And If you have any suggestion, you are more than welcome because we are quite naive users. And thank you all for your attention, and also to the group that where I work in Udine, and in particular Dr. Carolina Ricci and Eleonora Capezzali, who helped me with this data. And thank you.